Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Did you know there's a bustling, kind of secret, microscopic world inside us? One that might be shaping our health right from the very start. And what if I told you that tiny, invisible organisms living in our gut are surprisingly linked to really common childhood issues, things like allergies and asthma? Well, today we're taking a deep dive into exactly that world. We're exploring the critical role of the infant gut microbiome, specifically certain beneficial bacteria. We'll try to uncover how these microscopic inhabitants are just absolutely essential for immune system development. And maybe more surprisingly, how their absence seems connected to the rising rates of conditions like allergies and asthma in kids. So our deep dive today is built around a new health news article from July 4, 2025. And that article draws heavily from a pretty groundbreaking scientific paper. It's titled, Bifidobacterium Deficit in United States Infants Drives Prevalent Gut Dysbiosis, and it was published in Communications Biology. Our mission really is to distill what these sources reveal about how our early life experiences profoundly shape lifelong health. We'll want to give you the essential insights, without all the information overload. So let's maybe set the stage first, because the numbers here are pretty striking, aren't they? We're seeing this, alarming rise in non-communicable diseases, those chronic conditions like allergies, asthma, eczema, the ones that aren't spread person to person. And especially in children, global estimates suggest that maybe up to 40% of the population now has some form of environmental allergy. That could be pollen, dust mites, anobander. It's huge. And over the last few decades, we've just witnessed this growing prevalence of allergic conditions among children. Everything from mild seasonal sniffles to severe eczema, even life-threatening food allergies.
Yeah, and what's really fascinating, or maybe concerning, is that the emerging data suggests this rise actually begins much, much earlier than we used to think. We're talking about within a child's first 1,000 days. That's a really critical period. It covers their development inside the mother's womb and then right through the first 2 years of life. Now, environmental factors, lifestyle changes, they definitely play a role, no doubt about it. But scientists increasingly believe a key factor is disruptions in the gut microbiome. And in infants, this disruption often specifically involves the widespread loss of certain beneficial strains of bacteria. These are bacteria that are just vital for early immune development and ultimately long-term health.
Okay, so when we talk about those vital beneficial bacteria, is there one group that stands out? I know the paper focuses on Bifidobacterium. Can you tell us a bit about them?
Absolutely. Bifidobacterium are really central to this story. They're among the very first beneficial bacteria to colonize the human gut after birth, like the pioneers, you could say.
Yeah.
And their presence has been linked to a whole host of positive health outcomes. We're talking protection against metabolic diseases, against gastrointestinal tract infections, and inflammation, too. They seem to do a lot.
So they're not just hanging out, they're actively beneficial, like little health guardians from day one.
Exactly. You could almost think of them as tiny trainers for a baby's developing immune system. They're essential for guiding its development, teaching it to tell the difference between, say, harmful invaders and harmless things in the environment, like food or pollen. Without these crucial trainers present early on, the immune system might not develop optimally. And that could potentially set the stage for long-term health issues down the road.
Okay, that makes sense. So to figure this out, researchers launched this big study, right? The Baby Biome study.
That's the one. Yes, it's a significant investigation, a 7-year longitudinal study, which is important because it means they're following these infants over a long period. It wasn't just a quick snapshot. And it was specifically designed to fill some gaps in our knowledge because, honestly, before this, a lot of the studies on U.S. infant gut bacteria were pretty small, or they didn't really represent the diversity of the population here.
Right, so they needed a broader picture. How did they actually do it? What were they looking for?
Well, they meticulously analyzed the gut microbiomes of 412 infants, and these infants were carefully selected to represent the diverse demographics of the United States. They used some pretty advanced techniques. One is called metagenomics, that basically lets them analyze the genetic material of all the gut bacteria present. It tells you who is living in there. And then they used metabolomics, which looks at the compounds these microbes are producing. That tells you what they're actually doing, what functions they're performing.
Okay, so who's there and what are they up to? And the timing was important too, right? They collected samples early.
Critically important, yes. They collected fecal samples when the infants were between 1 and 3 months old. This period is considered absolutely crucial for early immune system development. And importantly, it's generally before the introduction of solid foods, which, as you can imagine, can significantly change the gut microbiome composition.
And what did they find? What was the big headline from this study?
The primary finding was, well, pretty concerning. A widespread deficit of Bifidobacterium in these United States infants. Just think about this. Approximately 1 in 4 infants, that's 25% between 1 and 3 months old, simply lacked detectable levels of Bifidobacterium in their gut.
Wow, 1 in 4. That's a lot of babies starting out without these supposedly crucial bacteria.
It is a significant number, and when they broke it down further, the deficiency was even more pronounced in babies born via cesarean section. About 35% of C-section infants lacked Bifidobacterium compared to 19% of infants born vaginally. So a noticeable difference there.
And what happens when those bacteria are missing? Does something else take their place?
That's the other concerning part, yes. In many of these C-section infants who lacked Bifidobacterium, their place was often taken by potentially pathogenic bacteria microbes that can sometimes cause problems. What's really interesting and a bit worrying is that some of these other bacteria can also utilize human milk oligosaccharides. Those are the special sugars in breast milk.
The ones meant to feed the good guys, right?
Exactly. They're essentially prebiotics designed by nature to nourish beneficial bacteria like Bifidobacterium. So if Bifidobacterium aren't there to use them, these other potentially less desirable bacteria can move in and thrive on that same resource. It's not just an empty space. It can get filled by the wrong tenants, so to speak.
That's fascinating. So it affects the whole ecosystem. Did they see other differences in the types of microbiomes?
Oh, definitely. The study also found that microbiomes that were rich in Bifidobacterium tended to have fewer genes associated with things we don't want, like antimicrobial resistance or genes that help bacteria cause disease virulence factors. These good microbiomes also showed more beneficial metabolic profiles. Their gut environments seemed healthier, producing more helpful compounds.
So they could actually group the infants based on their gut bacteria?
Yes, they did a sophisticated clustering analysis, which basically grouped the infants into 3 distinct gut microbiome types or communities. The first group, cluster 1, made up about 24% of the infants. This was the ideal group, you might say, rich in those beneficial Bifidobacterium that consume human milk oligosaccharides, especially a species called Bifidobacterium brave. These infants had better metabolic profiles, higher levels of beneficial compounds like indole-3 lactate, which helps modulate the immune system, and a lower, more acidic gut pH, which is generally healthier. And importantly, fewer of those problematic antimicrobial resistance and virulence genes.
Okay, so that's the benchmark, cluster 1. What about the others?
Then there was cluster 2, the largest group actually, at 37% of infants. This one was characterized by a high abundance of a different Bifidobacterium species, Bifidobacterium longum, and also another group of bacteria called Bacteroidota, kind of a mix. And finally, cluster 3, which was also quite large at 39%. This group was really marked by a scarcity of Bifidobacterium altogether. Instead, they had a higher abundance of bacteria from other groups like firmicutes and proteobacteria. One microbe, Clostridium perfringens, which can be a potential pathogen, was particularly abundant in this group.
And what did that cluster 3 profile look like functionally?
Not great, unfortunately. This third group had the highest levels of those antimicrobial resistance and virulence factor genes we mentioned. They also showed shifts in their bile acid metabolism, which can affect digestion and inflammation, and they had reduced production of essential micronutrients like thiamine. Plus, their gut pH was higher, less acidic, indicating a less healthy environment.
Wow. So 3 very different starting points for these babies' guts.
Yeah.
Now, you mentioned human milk oligosaccharides earlier. What about breastfeeding itself? Did that play out as expected?
This is where things got really interesting and maybe a bit counterintuitive. Generally, yes, breastfeeding was linked to higher Bifidobacterium levels, but mainly in the vaginally born infants. But here's the twist. In the C-section infants, the study actually found an opposite trend.
Opposite. Breastfeeding was linked to lower Bifidobacterium in C-section babies.
Exactly. In C-section infants, breastfeeding was associated with a decrease in infant Bifidobacterium. It suggests that in some specific situations, like after a C-section birth where the initial transfer of microbes might be different, breastfeeding might inadvertently allow other bacteria, potentially less beneficial ones, that can also use those milk sugars to colonize that niche instead of the Bifidobacterium. Remember that Clostridium perfringens I mentioned being high in cluster 3? Well, it was notably enriched specifically in C-section infants who were breastfed.
Huh. That really highlights how complex this interaction is. It's not just one factor, is it?
Not at all. It's a complex interplay between birth mode, diet, the mother's microbiome, the environment. It definitely challenges some of our simpler assumptions about breastfeeding always leading to higher bifidobacterium in every single case.
Okay, so we have these different microbiome clusters, these functional differences. But what does it all mean for the baby's health? How did these gut profiles translate into actual health outcomes later on?
That's the critical question, right? The My Baby Biome study followed up. They gathered survey results when the children were 2 years old, and those results showed that 30% of the infants overall had experienced at least one pediatrician-diagnosed adverse health outcome by age 2.
30%. What kind of outcomes are we talking about?
Well, looking at the specifics, 12.4% had developed allergies, 21.0% had eczema or dermatitis, and 3.3% had asthma by age 2.
So could they link the microbiome clusters to these outcomes? Was there a difference in risk?
Yes, they calculated the relative risk, and the findings were quite compelling. Infants who were in that mixed cluster 2 were 3.2 times more likely to develop one of these adverse outcomes by age 2 compared to the infants in the ideal cluster 1. And infants in cluster 3, the one really lacking Bifidobacterium, were 3.0 times more likely. So significantly higher risk for both groups compared to cluster 1.
3 times more likely. How does that compare to other risk factors, like, say, antibiotics?
That's a great comparison. The study noted that antibiotic use in the first 2 years of life led to a 3.3 times increased likelihood of developing an adverse outcome. So having a cluster 2 or cluster 3 microbiome profile carried a similar level of increased risk as early life antibiotic exposure.
Wow. And conversely, did having more Bifidobacterium actually protect them?
It did. The analysis showed that a higher abundance of beneficial infant Bifidobacterium overall was associated with a 3.1-fold reduction in the relative risks of these adverse outcomes. So it seemed genuinely protective. And when they drilled down into specific species, the presence of Bifidobacterium breve, that key species in cluster 1, was particularly protective. It was linked to a remarkable 4.8-fold reduction in relative risk.
Almost 5 times less likely. That's incredible for one type of bacteria. What about the other one, Bifidobacterium longum from cluster 2?
Bifidobacterium longum did show a trend towards reducing risk, but in this particular analysis, it wasn't statistically significant, so Bifidobacterium breve really stood out as the protective star here.
And did they find anything else, like specific microbial genes linked to the bad outcomes?
Yes, they did. They pinpointed specific genes associated with a higher likelihood of adverse outcomes. These included things like phage-associated genes, often coming from proteobacteria, and certain virulence factor genes, mostly from firmicutes, involved in things like immune modulation or bacterial defense systems. It's almost like finding the specific genetic tools within the microbiome that correlate with later health problems. Microbial fingerprints, as you said earlier.
It's incredibly detailed. Now, you mentioned some caveats earlier. It's important to talk about limitations in studies like this, right?
Absolutely. Transparency is key. And this study, while really strong, did have some limitations. For one, not all of the initial 412 infants provided the 2-year follow-up health data the analysis was based on, 210 who did. That's still a good number, but not everyone. There were also limitations in getting detailed medical records from all sites, so they relied on parental reports for the health outcomes, which can sometimes have recall bias, And finally, they didn't have super in-depth analysis of feeding methods. Things like whether the mother was a secretor, which affects milk composition, or the exact type of formula used weren't factored in, and those things could influence how an infant's microbiome responds to nutrition.
Okay, so those are important points to keep in mind.
Definitely. But despite these limitations, the study really provides a robust foundation. The evidence linking this early gut microbiome composition, particularly the lack of specific Bifidobacterium, to later health outcomes is very compelling. The term dysbiosis, meaning an imbalanced microbiome, can sometimes be debated. But I think this study, with its strong correlation between the lack of these key early colonizers and actual infant health issues, really points towards a genuine, impactful dysbiosis when these specific Bifidobacterium strains are missing in early life. It's not just different, it seems detrimentally different.
It really paints a picture, doesn't it?
Yeah.
Our modern lifestyle, C-sections, antibiotics, maybe diet changes over generations, seem to have fundamentally shifted this crucial early microbial environment. And this study strongly suggests these shifts aren't necessarily benign. They can be harmful, especially for infants. It's striking that even in vaginally born breastfed infants, getting enough of the right Bifidobacterium isn't a guarantee anymore.
Right.
You mentioned Bifidobacterium infantis was missing in 92% of these U.S. infants.
Yes, that specific subspecies, Bifidobacterium longum subspecies infantis, which is known to be very prevalent in infants in less industrialized societies and is a master utilizer of human milk oligosaccharides, was remarkably scarce in this U.S. cohort, found in only about 8%. It really underscores how much things may have changed in industrialized nations. The internal landscape seems different now.
So what's the big takeaway here? Where do we go with this knowledge?
I think the profound takeaway is that the infant gut microbiota represents this incredibly critical early window of opportunity. What happens in those first few months, microbiologically speaking, seems to have potentially lifelong health impacts. It highlights a period where interventions might have a really significant lasting effect.
It makes you think, doesn't it? Consider how this tiny invisible world inside us, shaped in our very earliest days, could hold a key to understanding, maybe even preventing some of the most common health challenges we face today, like allergies and asthma. What small shifts in perspective, or perhaps, future practices related to birth, feeding, maybe even targeted probiotics, might make a monumental difference for the health of the next generation? It's definitely something to reflect on. We encourage you to think about this knowledge, maybe how this deep dive changes your perspective on early childhood health and the importance of these invisible ecosystems within us all.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.